Hello, I'm Nolan Campbell, Global Medical Director for Tisabri at Biogen, and on behalf of the steering committee and other authors, I'll be presenting the study design and rationale of the NOVA trial. Natalizumab is a highly efficacious therapy for multiple sclerosis, but its use has been limited by PML risk. For many years, doctors have been experimenting with changing the dosing interval of natalizumab to see if they could improve the benefit risk by reducing PML and still maintaining the efficacy. And they've really been doing this in the absence of high quality data to let them know whether this was working or not. So earlier this year, we published work showing that natalizumab extended interval dosing, that is approximately 6-week dosing, is associated with a reduction in the risk of PML as compared to the approved dose. However, this was not able to look at efficacy. So in the absence of efficacy, we really don't understand the full benefit-risk profile of extended interval dosing. The NOVA study, which I'm going to be talking about today, is meant to address that gap. So in figure 1, you can see that we're presenting the overall layout of the trial design. And NOVA will enroll 480 patients that have been stable on the approved dose. And these patients will be randomized 1-to-1 to receive either 4-week dosing or 6-week dosing over the next 18 months. The primary endpoint is new or newly enlarging T2 lesions at 1 year. And we're often asked, why are you using a primary MRI endpoint as opposed to a clinical endpoint? And the reason is really for the dosing intervals that we need to study. Because we're looking at 4-week versus 6-week, if we ran this study as double-blind, we would require the patients to have to be infused every 2 weeks. And that's not really feasible for the patients. It's not good for the patients. So we're running this study as an open-label trial. And in an open-label trial, an MRI endpoint remains fully objective because you can have the raters for the MRI be blinded. Of course, clinical endpoints are also very important in this trial, and we're including relapse, EDSS, timed 25-foot walk, 9-hole peg test, SDMT as important clinical measures. We're also including pharmacokinetic and pharmacodynamic measures, with the idea being that even if extended interval dosing is slightly worse at a population level, we might be able to identify those patients who are doing particularly well based on their PK/PD profiles. If we look into figure 2, figure 2 is giving us the rationale for why we're looking at 4-week versus 6-week. And it's really based on the distributions that were seen in the earlier safety analysis, which showed a reduction in PML risk with 6-week dosing. So you can see that our dosing intervals that we're using in this trial are overlapping with the dosing intervals that were observed in the earlier touch analysis. If you look at figure 3 is giving the rationale for why we're looking at a patient population which has been stable on standard interval dosing for at least 1 year. And what we saw in the AFFIRM trial, which was the phase 3 trial of natalizumab, is that the efficacy of natalizumab increases after the first year. We know that the PML risk is very low for all patients in the first year. So we're taking advantage of the fact that PML risk is low over this first year and efficacy is increasing to give this trial the best chance possible for maintaining the efficacy while not compromising extended interval dosing as a possible PML risk mitigation. So in conclusion, this trial is going to provide the first randomized controlled data to assess extended interval dosing benefit risk and really test whether extended interval dosing is a possible PML risk mitigation technique for the future. What really motivates this project, and also is my personal motivation for being in this work, is to help identify patients on an individual basis based on their individual characteristics. What is the best treatment for those patients?